Hey everyone, welcome. Today we're diving into a scientific toolkit that is, frankly, incredible. It allows us to engineer life right at its very beginning. We're talking about embryo manipulation. And just so you know, these aren't some far-off ideas. These are powerful techniques being used today. So let's start with a puzzle. Take a good look at this animal. I mean, at first glance, yeah, it's a spotted cow. But what if I told you this heifer was born from two different embryos, from two completely different sets of parents? One set was brown, horned Jersey cows, and the other white, double-muscled Charolais. Which, of course, brings us to the big question. How on earth do you build an animal like this? It seems impossible, right? Well, stick with me and we're going to unpack the amazing science that makes it happen.
But before we get into the how, we really need to understand the why. What's the end goal here? Why manipulate an embryo in the first place?
Well, the source material is really clear about this. The prime purpose is, and I'm quoting here, enhancing the productivity of domestic animals. To put it simply, this is all about meeting human needs, for food and other resources by making livestock more efficient from the ground up. Okay, so here's the thing. No matter how wild these techniques get, everything we're about to talk about depends on two basic foundational procedures: being able to safely collect an embryo from a mother and then being able to transfer it into a surrogate so it can grow. These are like the essential bookends for everything else. All right, let's open up the toolkit. The first set of tools we're going to look at is all about making more, specifically how scientists can create clones and twins. So you've got two main ways to go about making a copy of an embryo. First, there's embryo splitting, which is pretty much exactly what it sounds like. And then there's cloning, which is a whole lot more complex. Let's break them both down. Embryo splitting is the most direct method you can imagine. A scientist, using these incredibly fine microscopic tools, physically splits an early-stage embryo right down the middle. Each of those halves now has the potential to grow into a complete, identical individual. And the result? Well, it's pretty much what you'd expect: monozygotic, or identical, twin calves. They're born from that one original embryo that was split and then put into a surrogate mother. It's a really effective way to double the offspring from a super valuable embryo. But there's always a catch, isn't there? This technique has its limits. While you can split an embryo, the survival rate drops off, and it drops off fast, the more you divide it. So if you split a 2-cell embryo, each half has about a 30% chance of making it to birth. But if you wait until it's at the 8-cell stage and then split it, that rate just plummets to 11%. There's a real biological cost to each division. So you can see the problem, right? Splitting only gets you so far. That brings us to the much, much more complex alternative for making copies: cloning, through a process called nuclear transfer. Now, I know this diagram looks a little intimidating, but the core idea is actually super elegant. You take an egg cell and you remove its genetic material, its nucleus. Then you take a cell from the animal you want to clone and you fuse it with that empty egg. Let's walk through how it actually works. So first, you remove the chromosomes from the recipient egg. That step is called enucleation. Next, you place a cell from the donor embryo right up next to it. And finally, you use a tiny electric current to fuse them together. That little shock basically tricks the egg into thinking it's been fertilized, and just like that, a new, cloned embryo starts to develop. Okay, so making copies is one thing, but what if you want to get more selective? That moves us to the next part of our toolkit: picking and choosing designer traits, like the sex of the offspring or specific genes.
Yeah, this is where we go way beyond just copying. These techniques let scientists interact directly with an embryo's genetic code. And think of it like this: there's a huge difference between just reading the code to find traits that are already there and actually writing new information into it to add brand new ones.
And here's what those two approaches look like. On the left, you've got embryo sexing. That's our reading tool where you're just identifying a trait. On the right, you have gene transfer. That's the writing tool where you're actively adding a new trait. So how do you actually read an embryo's sex? It's a really cool process. First, they take a tiny biopsy, just a few cells. They get the DNA out of those cells and make lots of copies of it. And here's the clever part. They add a special probe that is designed to bind only to the Y chromosome. If the probe latches on, you know the embryo is male, XY. If it doesn't find anything to stick to, you know it's female, XX. Simple as that. So we've covered making copies and selecting traits. Now for the final and maybe the most mind-bending tool in the whole kit: mixing and matching different embryos to create something called a chimera. So what in the world is a chimera? Well, in biology, it's a single animal that's actually formed from the cells of two or more completely different embryos. The result is one organism that's made up of a patchwork of different genetic tissues. It's not a hybrid, it's a mosaic. And this is the answer to that puzzle we started with. That impossible cow is a chimera. It was made by fusing two early-stage embryos together, one from those white, double-muscled Charolais parents, and the other from the brown, less-muscled Jersey parents. So those brown and white spots, they aren't just a color pattern, they are literally patches of tissue from two different sets of DNA living in one body.
But you might be asking, why would you go to all this trouble? Well, here's a perfect example. That double-muscled trait from the Charolais is great for meat production, but it comes with serious problems, like very difficult births and calves that need a ton of extra care. By creating a chimera with a normal embryo, scientists can actually moderate those negative effects. You get the benefit of more muscle without the severe health issues.
So let's recap. We've got tools for making more, tools for picking traits, and tools for mixing them together. What happens when you combine that whole toolkit? Well, you start engineering the future.
This diagram really shows you just how incredible this all gets. You could, for example, start with one valuable embryo, figure out its sex, then split it to make a twin, and then transfer a specific gene into just one of those twins before implanting them. These techniques can be stacked and combined in all these complex ways, and it's opening up a whole new frontier in biology.
And all this leaves us with a final massive question. We now have a toolkit to combine, to copy, and to edit the very building blocks of an organism. Now that we can truly engineer the beginning of life, what's next?